Welcome to Mitosis Square Garden. Tonight we have wrestling chromosomes that will take us through the mitotic cycle from interphase through cytokinesis. The match begins in interphase. The wrestlers have just entered the nuclear envelope wrestling ring and are ready to prepare for the mitotic cycle. The two centrosome referees are standing outside the nuclear envelope getting prepared for the division of Mr. Orange and Mr. Green chromosome. The wrestlers went through the beginning stages of interphase all by themselves until they realized they had to call for backup. They were getting nervous for prophase, so each chromosome called on his identical twin to help through the rest of the mitosis fight. The wrestling chromosomes and their replicated twins enter the prophase. The wrestling chromosomes are becoming more condensed. The centrosome referees are getting their spindle ropes ready outside of the nuclear envelope ring. The fight inside the ring is getting so intense the sides of the wrestling ring nuclear envelope are starting to fragment. The referees run onto each side of the nuclear envelope and start to open their spindle ropes. The chromosomes have now entered the longest stage of the mitosis fight, which is called the metaphase. The wrestling chromosomes are stuck in the spindle ropes on the metaphase plate between the two referees. The spindle ropes are tied around each wrestler. Finally, the centrosome referees are strong enough to start anaphase, in which they pull the wrestling chromosomes apart from one another. The kinetochore motor molecules walk along the spindle ropes, pulling the wrestling chromosomes apart. The chromosomes move towards opposite sides, and their kinetochore microtubules become shorter. The mitosis fight is almost over as the chromosomes enter telophase. The wrestling twins are separated, and the two parts of the chromosomes are now two genetically identical diploid chromosomes. The referees' centrosomes on opposite sides each release their spindle ropes from their chromosome wrestlers, knowing they cannot reattach since a cleavage furrow has formed. The contraction of the actin and myosin, which make up the microfilaments, create the cleavage furrow. Now that the fight is over, the nucleoli reappear, and the wrestling ring nuclear envelopes start to be rebuilt on each side of the cleavage furrow. Cytokinesis finishes the mitosis fight, the cytoplasm divides, and two identical daughter cells result.
Welcome back to Meiosis Stadium, where your home team, the chromosomes, are down by one run here in the bottom of the ninth. As the bottom of the ninth begins, interphase starts. Leadoff batter, homologous chromosomes, stands in the on-deck circle as the cell replicates its DNA. The chromosomes condense in prophase 1. Each of the chromatids go through the process of crossing over, in which enzymes slice sequences of DNA and exchange genes. They all high-five at their chiasmata, the points where crossing over occurred. Yelling divide, they separate slightly. Metaphase 1 begins, and homologous chromosome steps to the metaphase plate. The centrosomes move to the opposite poles of the cell. Both chromatids attach to the kinetochore microtubules, while the others attach to non-kinetochore microtubules from the opposite pole. The pitcher winds and throws the ball in anaphase 1, caused by a protein breakdown. The batter swings as the homologous chromosomes separate. Keeping both of his hands on the bat throughout the swing, his sister chromatids remain attached. The batter hits the ball as telophase 1 begins. Each half of the cell has a complete haploid set with two sister chromatids. He rounds first as cytokinesis occurs. Two daughter haploid cells form along the cleavage furrow, and the batter ends up on second with a cell double. As the ball is thrown into the pitcher in prophase 2, a spindle apparatus forms. Centrioles separate, and a nuclear membrane fragments disperse. Chromosomes in the two newly formed cells step to the metaphase 2 plate. The sister chromatids are not genetically identical. The pitcher delivers the ball again in anaphase 2. The breakdown of proteins allow the chromatids to separate towards the opposite poles, and the batter swings. The ball flies towards the gap, and nuclei form. Cytokinesis occurs, and the batter and the one on deck end up hitting doubles. The three doubles lead to four daughter cells with haploid chromosomes, and the two runs lead to a genetically distinct win. Mitosis has one division, while meiosis has two. Crossing over occurs in meiosis, but not in mitosis. Mitosis results in two identical diploid daughter cells, and meiosis results in four genetically different haploid cells, each with half as many chromosomes as the parent cell.